All right, so good evening, everybody. We are here on week 4 and talking about how cells work. Tonight, our plan is going to be to look at our Blackboard and see where we are with adding new material. And then I want to go through the lab folder, and then there's a PowerPoint that I've put together that I want to go through with you so that you can see step by step how you need to use the PowerPoint, the lab manual, and the activity sheet all together. And we can talk about how that went last week for you and maybe some ways to make that less cumbersome. I know it's definitely cumbersome. It wasn't our first choice for how that should look, but we tried something new. So we're going to go through all the lab material. I have one animation that I want to show you from the PowerPoint, and you'll see the PowerPoint is up. Last week we had talked about me doing the narrations for the PowerPoint instead of Miss Shepherd doing those, and I recorded it, but the closed captioning takes so long. So I know that's not really an excuse, but I've got to figure out how to do that faster. So it'll take me a whole day to do the closed captioning for this, and then another day to do the PowerPoint. So anyway, there's a lot going on, right? We all are trying to figure out how to best manage that time. I also want to introduce to you our semester project. Get into it, you'll see it. I think it's going to be pretty cool. And that should be about it. So if you have questions along the way, feel free to unmute yourself at any time and ask those questions. Or if you want to put something in the chat box, that'll be just fine. So I'm going to go ahead and share my screen, and we will look at we'll look at the Blackboard together, and then we'll go through and look at different parts of the Blackboard. So if you have questions, let me know. Otherwise, I'm going to go ahead and get started with. Okay, let me put this here. Okay, so here we are looking at our announcements. Hopefully you've seen, I've posted 2 announcements so far this week. Yesterday I posted about tutoring opportunities. So if you are finding that you're having a difficult time with concepts or with studying, with organizing information, please reach out to the STEM Learning Center. The two of the 3 biology folks I know very well, and they're just phenomenal people. So they're great to work with. Not only are they professionals in their field, they're just phenomenal and they love what they're doing. So I highly recommend, if you're coming across any issues that you're not sure, that you want to reach out to either me or your lab partner. I think Cruz and Rachel, aren't you 2 lab partners? I think so.
Yes, we are.
Yeah, okay. So hopefully you guys have been in communication a bit and able to lean on one another as well as encourage one another. Because now in week 4, we are in it. We are in it to win it at this point. And then there's also, and so the STEM Learning Center, those tutors are all here at FCC. Thinking Storm is a third party that we contract with. And I think it says you get 15 hours. So that's across all of your different subjects. You have 15 hours. And what's cool about them is that you can send them a lab report or a paper that you're working on, and somebody catches it and they will get back to you and they'll have your paper edited for you the following morning. So if you sent that out at 1 o'clock in the morning, by 10 a.m. when you can be in touch with them again, someone's already looked at it. It's a really cool system. I've had positive feedback about it anyway. So on Sunday, I sent you a very late night Sunday update. So what you'll see when we go into our course folder is that you have the PowerPoint is now with the lecture notes. It's not narrated. I'm going to try my hardest to get that done for you. And then there's the chapter 5 quiz that you have the 2 attempts, 25 minutes, and folks have been seeming to do pretty well with those. So that's encouraging that you're everyone's feeling pretty comfortable with the concepts. And then you'll also see in the lab folder, just like last week, we have a PowerPoint that goes along with your lab manual and your activity sheet. And you have that activity sheet, the table assignments, the same thing. Then you upload that to your post lab. And instead of this week having an additional assignment that is a worksheet or a test or a quiz, we have our second of 6 discussion forums. So I'll talk a little bit more about that when we get into that folder. Like I said, I'm going to introduce the semester project. So here are all of your due dates. And my folks who are here, you have checked in for your synchronous session. So welcome to that. Next Tuesday, we will be having a synchronous session, and it's going to be really focused on our lecture exam and making sure that you're ready. So we'll have an exam review in our next synchronous session. And then Thursday, you're going to have your initial post is due in the forum, just like the first one. And then by Sunday, you'll have 2 responses due, just like the first setup that we did. So hopefully you're going to see that we follow the same patterns and feeling more comfortable with those. And again, on Sunday, your chapter quiz is due, your lab is due, and then your discussion forum will be due. Okay, so let's dive into the material unless you have questions right now. Okay. All right, so when you open up week 4, you'll see here's our Zoom link. And then our lecture 4, we have, here's our PowerPoint and the quiz that goes with it. So I'm going to pull up the PowerPoint on my other screen, and then I'll share that with you. So give me just one moment to pull that together. Okay. All righty, so are you able to see my PowerPoint right now?
Yes.
Okay, great, thank you. Hi, Carson, welcome. So when you are looking at these PowerPoints, please make sure that you have this view so that you can see not only my PowerPoint, but the notes that I put in here for you. Those are super important. So, you know, in here, I'm saying, you know, please make sure that we're talking about how cells work. Refer back to that chapter 1, you know, the 6 characteristics of living organisms, and make sure that you know those 6 pieces and start thinking about how does that relate to how the organelles work that we worked with last week. And then this week, when we're talking about photosynthesis and cellular respiration, how do these characteristics, how they evolve, and how are they important for understanding living organisms. So please make sure that you're really looking at these notes that are down here as well. There's the objectives. Okay, so I want to talk a moment about the photosynthesis and cellular respiration cycle. So when we're looking at that cycle, it truly is a circle. So when we're looking at photosynthesis, we have, if you can see down here, our inputs, our reactants in this chemical reaction are carbon dioxide, water, and sunlight. So that's what's going into the chemical reaction. On the other side, our product is going to be glucose and oxygen. Okay, so the input is carbon dioxide, water, and sunlight. The product is glucose and oxygen. So if we come down here, exactly what our products are with that, with photosynthesis is what our cellular respiration means for that process to work. So cellular respiration means glucose and oxygen as its reactants and goes through that chemical reaction, and our products are CO2, H2O, so carbon dioxide, water, and energy, where if you go right back around, that's exactly what we're talking about. Okay, so we need the reactants to be the products for the photosynthesis cycle to work, and we need the reactants of photosynthesis to be the products for cellular respiration to happen. If the cell is not creating this ATP, this energy, it will die. So this is the most critical process in biology, because this is what's keeping all of the cells alive. And that energy is being created in the mitochondria of the cell. If you remember the mitochondria from our organelles last week. Okay, so I just want to make sure that you understand how those cycles are really feeding one another. Does anybody have any questions on that one? Okay, so I'm going to move on. And I won't go slide by slide for this. I'll let you do, go through it and you can read it. And if you have questions, just let me know. Here's that word biomolecules, if you remember that from last week. And those biomolecules, here we have our monomers, and those are the pieces that make up the chain of polymers. So what we can see is that this is a protein. Yeah. Okay, here's another way of looking at how those cycles really feed into one another. And this sugar right here, that's really feeding the energy sources, because that's what's being broken down and made into energy. The oxygen that goes in with the sugar, that is helping to release the energy. But those are not steps that you have to really get into. What I really want you to understand is what this cycle looks like and how it functions and relies on each other. Okay, so another piece that I want you to know about are enzymes. And enzymes are what speeds up a chemical reaction. An enzyme is a protein, and so it's made out of different chains. And there's all different kinds of enzymes that do have different kinds of functions and structures. So as long as an enzyme is working normally, then what you're going to find is that it can do what it needs to do to speed up a chemical reaction, and then it's going to go back to its original structure, unless it's denatured. And what that means is that that chemical structure is broken. So it doesn't actually go anywhere. The enzyme's still going to exist. It just means it's not going to be working anymore. So with this chemical reaction, we want to see So without the chemical reaction, that's another one of those game changers for life, is that chemical reactions are going to happen, but if you leave them to themselves, they're going to happen slowly. So the purpose of having an enzyme is to make a chemical reaction happen fast so that we're able to have life. So that's another big one to understand, is the purpose of enzymes. And again, those are made out of proteins. We have a lab coming up called, what is it? I want to say it's pineapples and jello, but that's not, I don't remember the actual name of it. But we're going to be talking about denaturing enzymes. So this is a concept we get to put into application, and it's a really fun lab. So I think we'll enjoy that one. Okay, questions about the enzymes and their importance in what's happening inside of the cell? Okay. Alrighty, so I'm going to move us forward. Okay, ATP. So like I said, this is the energy that all cells need, all living cells need to continue to live. So ATP is adenosine triphosphate. So it is one of these adenosines and three of the phosphate molecules that come together. And what happens is when we're going through, when energy changes, because if you remember, the first law of thermodynamics is that energy cannot be destroyed. So here, it can't be created or destroyed. So here we have, as ATP is going around, we might lose this phosphate because it has another job to do. And then we have ADP. And as we have energy from the sun or glucose coming in, we're going to collect another phosphate, which creates the ATP. So as we're looking at it, here we have cellular respiration is happening here. On this side, we have photosynthesis happening on this side. So in the cellular respiration, we're losing one of these phosphorus atoms, and then in the photosynthesis process, we're gaining a phosphorus atom. And we'll talk about why that's significant in a moment. Okay. So here we have our chloroplasts. All right, so our photosynthesis is happening in two stages. And this is where that ATP that we were just talking about becomes important, because it's coming from this light reaction. It's going to be going into the Calvin cycle. And that Calvin cycle is what's going to be releasing the sugar. So if you remember the photosynthesis, the products are going to be glucose and oxygen. So that's what we see. We have the CO2 coming in, we have energy coming in, goes through this next cycle, and we have sugar and oxygen coming out on the other side. So, and you can see that here. So our ATP goes in here, and then we lose, there's that phosphate that we lose. And I don't want to go too far into the chemical reactions within photosynthesis and cellular respiration. You really don't need to know all of those specifics. I just want you to understand that these chemical reactions have atoms that are flying in and out, because we've been talking a lot about about the mobility of atoms and how it works with water. The importance of when temperature is heating up and when it's cooling. So it's important to understand that all these processes have chemical reactions happening within the cell, and it's the atoms within the cell that first biological hierarchical level is functioning, and that they have jobs to do. So I don't want to harp too long on it, but it's really All righty. Okay. Okay, so now what I want to show you is an animation about photosynthesis. So let me go find that for you. Does anybody have questions while I'm finding that? Okay. All right, and are you able to see my screen? It should be opening up a video here. Yes, thanks, Bruce. Okay, great. So let me see. Let me know if you can see.
Eukaryotic cells get the bulk of their usable energy in the form of ATP by the multi-step process of cellular respiration, which takes place partly in the cytoplasm and partly in mitochondria. During cellular respiration, the carbon-carbon bonds in glucose molecules are broken, and each carbon atom is released into the environment in a molecule of carbon dioxide. Water is a byproduct. Cellular respiration is aerobic, meaning that it requires oxygen. By the end of cellular respiration, glucose is completely broken down and the cell has gained a large amount of ATP. Cellular respiration can be broken into 3 stages, the first of which, called glycolysis, occurs outside mitochondria in the cytoplasm. During glycolysis, sugars, mainly glucose, are split to make a 3-carbon compound called pyruvate. 2 ATP molecules result from glycolysis, 1 for each pyruvate formed. In addition, glucose provides high-energy electrons that combine with NAD+ and hydrogen ions to make 2 molecules of NADH. NADH will be important in the final stage of cellular respiration. Although glycolysis itself does not require oxygen to run and is therefore considered anaerobic, most eukaryotes add 2 more stages to the breakdown of glucose, the Krebs cycle and oxidative phosphorylation, which are aerobic. These last 2 stages are interdependent and only run when oxygen feeds into oxidative phosphorylation. After pyruvate is produced from glycolysis, it enters mitochondria, where it is broken down in a sequence of enzyme-driven reactions known as the Krebs cycle or citric acid cycle. Notice that the mitochondrion has 2 membranes, the outer and inner mitochondrial membranes, which establish separate compartments within the organelle, the intermembrane space, and the matrix. The Krebs cycle takes place in the matrix. In the Krebs cycle, the carbon backbone of pyruvate is completely taken apart, releasing carbon dioxide that enters the atmosphere. The breakdown of carbon backbones produces a large bounty of energy that can be used to produce several types of energy carriers, including ATP, NADH, and FADH2. ATP is the common energy currency of the cell and can now be used. NADH and FADH2 are converted into ATP in the final stage of cellular respiration, oxidative phosphorylation. Oxidative phosphorylation provides the largest output of ATP during cellular respiration. It occurs in and around the inner mitochondrial membrane. In the membrane, several protein complexes form an electron transport chain, and another, ATP synthase, forms the bulk of ATP from cellular respiration. The energy carriers NADH, shown here, and FADH2, not shown, begin this final stage by donating high-energy electrons, originating from glucose, to the electron transport chain.
Okay, I'm going to stop it there for just 1 minute. So this ATP synthase, this is where the enzyme becomes important. So anytime you see this ASE, this A's, it's usually going to be an enzyme. And when you see something like glucose, fructose, sucrose, that's going to be a sugar. So let this ending here help you to determine that we're talking about an enzyme. So that's the enzyme that's helping the ATP move in and out of the cell, and they have the third step. I want you to consider that just call it the electron transport chain. we don't need to go into all of these details, but just know that the electron is traveling through. So it's a chain, it's all linking together. Okay, I'm gonna, any questions to this point in the video? Okay.
These electrons are passed from one complex to the next until they get to the end, where oxygen molecules grab the electrons, combine with hydrogen ions, and form water. Oxygen is required in cellular respiration as a final acceptor of electrons, and water is a byproduct. Note that NAD+ is released into the matrix after donating electrons to the electron transport chain. It is now ready to accept additional electrons from glucose. What does this chain do other than transport electrons to oxygen? Members of the chain also act as proton pumps, so as they transfer electrons, they also pump protons, also called hydrogen ions, across the membrane. In this process, electrons become depleted in the matrix as they are pumped to the intermembrane space. A concentration gradient of protons thus forms across the membrane. When one side of the membrane has a high concentration of protons and the other has a low concentration, protons will tend to flow back across the membrane, equalizing the concentrations. They flow through a channel in the ATP synthase complex. ATP synthase uses the energy of their movement to combine ADP and phosphate into ATP. In this way, the chemical energy of NADH and FADH2 is converted into the chemical energy of ATP. In this process, a large amount of ATP is generated. In fact, oxidative phosphorylation can generate 15 times as much ATP as glycolysis alone can.
So why that's significant is that when we're talking about prokaryotes do not have a mitochondria. So prokaryotes are only going to go through glycolysis. Whereas they, in the eukaryotes, they have the mitochondria organelle, so they're going to have that output of the 32 ATP, which is going to give that cell and all of the organelles more energy. So that's really, that's an important part to understand. So the prokaryotes are going through cellular respiration, but they only have that first step. They don't go through the second two steps because they do not have the mitochondria. Okay. Okay. So then the rest of this, I think you can go through and talks a lot about photosynthesis and changing light. And here, this is a diagram that's out of your book. This is one that you're going to want to mark for your discussion forum that you can refer back to. And we'll talk a little bit about that. So this is fermentation, which is cellular respiration without oxygen. So an anaerobic process. Okay. And then you'll see as you go through, here are your clicker questions. So just like those reading quizzes that you take for lecture, these are really important for you to review. So I'm going to leave this, and then there's 10 of those. I'm going to leave this PowerPoint now, unless you have some questions. Okay, so we'll leave that where it is. And now I am going to change to look at okay, so here we are looking at our Blackboard site again. Okay, so that's everything that you'll find and need to look through in your week 4 lecture folder. When we go back out and we come here to our lab 4, paper chromatography fermentation. So this is a 2-part lab, and I am very excited to to share this with you. Has anyone had a chance to look this lab over? Not just yet. Yep, I get it. I just turned in the other one, right? Okay, so I'm going to share my screen with you. There we go. All right, so now we are back to looking at the PowerPoint that you'll find in your lab. So I gave you some background about chromatography and what that is. And so what we're doing is we're separating mixtures and we're separating them from one another. So part of your questions is what's happening with black ink? Is black just going to stay that color? Well, black is a combination of many pigments. So, you know, what does it look like as those pigments are going to separate? And what can we understand about those compounds? Because they're going to go to the same place every time. What does that mean? So when we look at the phases of chromatography, we have two. So the first phase is our stationary phase, and this is when you put that initial black line on your coffee filter. I will also say that if you do not have a coffee filter, you can also use a paper towel. But I caution you with that. So the paper towel, you don't want to have other colors on that paper towel, so you want just the plain white kind. And the super absorbent is not very helpful. So if you have like a cheapy, you know, a cheapy paper towel that you save for cleaning the windows, pull that out, and that would be a good alternative if you do not have a coffee filter. Okay, so sorry, I digress. So we go back to these two phases. Here we have our stationary phase, which is where that substrate, either the coffee filter, I'm just going to call it the coffee filter. The coffee filter is holding on to that ink. And so that liquid is staying put. That's phase one. Now, when we add a solvent, which is your water, to the bottom underneath of that line, you're going to see that the wicking of that water is then going to have a chemical reaction that's separating the pigments. And that's what we're going to be measuring. So let me continue on. I'm going to skip forward to this so that you can have a nice visual of what your setup's going to look like. Now, instead of just doing a dot, ours says to do a line, and I think you'll see the separation of pigments a lot easier with the line versus doing the dot. Okay, but you're going to be able to see here that we do have a lot of different pigments that are being pulled out of this black line. Okay, so this is the setup. You have your glass of water, you have your solvent, which is going to be underneath of the filter. You put your coffee filter into it, and then it comes all the way up. Let's see. When you measure that's a great question. I will answer that in the next slide. Just give me just 1 minute. And you are right on track with your question. Okay, so then I'm going to back up here to slide 2. You can't see, can anybody see the slides? Anybody? Come on now.
I can see them now.
Okay, good. I have to click the button, the share button for it to work. Thanks for letting me know. My apologies about that. Okay, so let me pull this back over. Okay, so I'm going to go back up to the top and just show you what I've been talking about here. So this is the beginning of your slide of your PowerPoint here, and I pulled us forward just to slide 3. And so this is the visual setup that you'll have at home. And so instead of making that dot, make a line, the 2 centimeter line from across and then from the bottom 2 centimeters. So if we look back up here, before you put water into your glass, you're going to want to hold you're going to want to make sure that your water does not go over your line. If the water goes over your line, you're going to see the diffusion of that water, of the ink down into the water. And that's not what we want. We want to make sure that the water is underneath of the line so that the coffee filter will wick the ink up instead of going down into the water. Okay, so I just want to make sure that you see, so you want to know where that line is, add the water, and then put in that coffee filter. Okay, so cruise. This is where we're headed. So we have this retention factor. And our retention factor is looking at compound chemical pigment specific. Of how far is that solvent going to cause that pigment to move? And what we're going to see at the top of our colors, we have this. It's called the solvent line. And that solvent line, when we hold up a ruler next to our glass, it comes out to 4.7 centimeters. So we're going to put that, the distance that the solvent moved all the way from the bottom up to the top, we put that on our, whoop. Are you able to hear me? I'm seeing that my internet is unstable.
I can hear you.
Okay, great. So you have your denominator is your solvent, and then your numerator is going to be how far did your pigment move. And so you'll actually see that dot move up, and you'll see the lines move up the coffee filter, which is really cool. So if we're looking at A, this first dot, it comes in at 0.7. So you put that over in your numerator, and then your retention factor is going to be this 0.15. And same goes with B and C. Now, if you get a number that is greater than 1, greater than 0.99, we've probably switched the solvent and the ink spot. So make sure that your number is under 1, is less than 1. Okay. All right. So why is this something that we're interested in? And how is this? So Cruz, does that answer your question? Okay, just let me know if you have, oops, sorry.
The question that I had, when I did the lab and I put the coffee filter in, I started to bleed up. When I looked at the actual lab manual and then did the activity, I couldn't tell where they were starting to measure, if at the beginning of the bleeding, the middle, or at the where the bleeding actually stopped and turned a different color.
That's a great question. So you're going to go at the top of that pigment, and then before it goes to the next color.
That makes sense.
Okay. Okay. Yep. So you're going to go right at the top of wherever that pigment stops, and then it's going to, and then when it transitions, you go to the top of that pigment. So you tried it. What did you think? Did it work out?
It was interesting to just watch all the colors separating and getting away from each other. And I didn't know that was possible, but there it was.
Well, good. I'm happy that it worked out for you so you could see it. So why is this important? Is that if we have an unknown compound, we can measure a known compound with that unknown compound and start to infer what we can from this, comparing the known to the unknown. So if I know that this right here, B, is carotene, that's the chemical compound that has the retention of 0.87, and then I test something else next to it and I'm like, oh, it has the same. Okay, so I'm guessing that this is going to be the RF factor. The value is the same. I'm going to make a guess that this is carotene, and so that it gives you a comparison. Now, that said, every sample is also going to be different, so there's definitely room for error. I would say that it never comes up, 8.7 is carotene. There's always a little bit of wiggle room in there, which is when we want to start using some more technical equipment, but that technical equipment is in our lab space that we can't access right now. All right, so you'll also see in your lab where you're comparing different spinach leaves and fall and summer leaves. So I wanted to show you what it would look like actually doing that, because the simulation that you do, it doesn't show you like, what does that mean? So you've taken these leaves and you've let them diffuse into this water, and then that pigment is left in the water, and so that's what you're measuring, so that what pigment is left that diffused into the water. Okay, so I'm going to take us back to the lab itself. And so, this is your lab manual, you're following through. Here's your retention factor. Here's the part that you're doing, your kitchen lab. You're making your predictions, you're figuring out RF factors, and you're writing those down here. And so these tables you find also are in your tables and activity sheet. Okay, so you're filling that in, because you've got your 3 different things all happening at the same time. You've got your lab, you've got your PowerPoint, and you've got the activity sheet. So I'm back in my lab, I'm going through, and then I get to activity 2. All right, so I'm going to click on this activity 2. There we go. And when you click into it, it should go directly to this page. If it doesn't, along the tabs here at the top, you're going to see Simulator. Just click on that tab. This is what should come up for you. All right, so I want to play around with this for a moment so that you can see what that looks like. Here we have our plant pigments. So you have in your lab, it says that you're going to either use spinach greens, you're going to use the maple leaves in spring, summer, autumn, winter, or the fall foliage. So that's where you're going to find which substrate you're working with. And then here, your solvents, you're either going to use acetone or alcohol. And I believe for all of ours, we use the acetone. And all you have to do is hit start. And then you watch our make-believe spinach leaves going up the filter. Separating. Okay, so we're going to click on this test tube to find our RF values. So before they give you that information, you have to identify the band. So where's that information for you? You have to go back to your lab manual. Okay, and you're going to have this table 1 has your pigment and has your chemical, and then here it has the color that you're looking for. So if we are looking for, what color are we looking for? So it's asking us to look for the yellow band. What is the yellow band when you're looking at the table? Can someone tell me, please? Go back to here. Look at table 1. Which pigment are you?
Is that like the one that starts with the X? I can't pronounce that.
Yep, xanthophyll. Good. So then we come back into here and says select the pigment identified. Drop down. Okay, good. Those are all the same ones that we were looking at in the table. And oh, sorry about that. So Cruz, this goes right back to what your question was. I was looking at the yellow because it's touching the yellow. It wants you to look at the top of the light green. I apologize. So if we were looking at the xanthophyll at the top of the yellow, this little arrow would be pointing up here. So we're really looking at the top of the light green. So which one is it? Would we say chlorophyll A?
Yes.
Yep, all right. So let's go and give that a try. Okay. Now it's doing mine.
I think you're gonna have to pick the one with the letter X.
Oh, did I not do that before? Jeez. Sorry, guys. Ay, ay, ay. So, okay. So it's easy. It's easy. It's just like that. My apologies. All right. So then what's going to happen is that these two squares light up for you. And it's going to show you your scale. So you get to see your measurements. And then, so you can figure out what your RF factor is. So that your first factor, so you have 6, so 6.3, if we called that 6.3, that would be our solvent denominator, our solvent factor here. And then looking at this yellow then, we're going to be 4. Would you agree? 4.4. So then that would be your numerator, and you divide your solvent into this measurement here. And then that's going to give you your RF factor. Now, here's the best part. Show the chart. Ta-da! There you go. Your RF values are all figured out there for you. So what you're going to be doing is you write this and you put it right into your lab. So you calculate your RF factors and then compare them to the computer calculated RF factors. So do take the time to calculate your RF factors so you get some practice with what that looks like. And then you know you're going to have the answer there so that you can have that comparison. And then you have questions here about why you might see some changes. Okay, so then what's going to be neat is that you can go back into this program and try a new, try the maple leaves. And you're going to see that separation of the pigments and looking very similar to the spinach leaves. And then you're going to compare that to the fall winter leaves. So have some fun with that. I think you'll find once you get in there, this part is not going to take you very long. Okay, so that's really all activity 2 is in your lab this week. Do you have any questions about that? Okay, so now we're going to get into part 2 of your lab. And this is when we're talking about fermentation. So here we go with our anaerobic cellular respiration, yeah, that we were just talking about. Now, the difference here is our anaerobic cellular respiration, or I'm sorry, our aerobic means that we have oxygen. It uses oxygen. Fermentation, there is no oxygen here, so it's an anaerobic process. The AN means without. So what's going to happen to the sugar on this other side? We're going to have alcohol. Yeah, so this is our ethanol over here. So we're not getting that water on the other side. We're getting alcohol on the other side. Okay, so for your lab, you're going to have your activity sheet out. Okay, and you're going to, this is not it. Okay, here we go. So you're going to fill out this sheet based on your observations from the PowerPoint. Okay, so that's just like it was for experiment 3. So then when we go back to our PowerPoint, oh, and this is a shout out to my elementary ed folks, that you can have fun with chromatography with little guys too. Okay. All right, so we want to know, does temperature have an effect on the rate of fermentation? So remember, fermentation is the breakdown of glucose without oxygen. So is temperature going to affect the rate at which that glucose is being broken down? So what we're going to have, we're going to be looking at the input of sugar, what happens when we change the temperature, and what the output is going to be. And we'll see that we have that carbon dioxide and alcohol in our 2 energies. So this is also something to really consider here, is when we have the oxygen, on the other side, we get all of the energy. The mitochondria is able to complete its process and produce 36 ATP. When there is no oxygen, this shouldn't be here. When there is no oxygen, we only get 2 ATP. My apologies. Okay, so this is what our test tubes are going to look like after we add in. So I want to think about those questions. That's what our test tube is going to look like when we have our yeast and our water mixed together. So step 1, we have 3 different beakers, 3 different glasses with 3 different temperatures. So we have 20 degrees, which is room temperature. We have 40 degrees, which is going to be that heated, like bathtub temperature. And then we have chilled, which is right below or right above freezing at 2 degrees. Next, we're going to set out 6 test tubes. 2 of those test tubes are going to have water for room temperature, 2 have for the bath water, and 2 have for that cold water. Only 1 of test tube in each of those groups is going to get sugar. So let's look at this. So test tubes 1, 3, and 5 all have the yeast, the sugar, and the water in the test tube that are going to be activated. In 2, 4, and 6, you're only going to have yeast and water. So remember, we really want to make sure that we have that so glucose is what the yeast is going to be breaking down. Okay, so it's important that we recognize the value of having the sugar in the test tube. So thinking about it from an independent and dependent variable outlook here. Okay, So we shake up all 6 of our test tubes. We've put a balloon on top of it. We're shaking it up to activate the yeast. And now we flip the test tube, and all of the yeast, sugar, water mix, that solution goes into the balloon, and you tie the balloon off. Okay? So now we're going to put that balloon into their water, into the temperature of the water that we put into that mixture. So we will see that we have 2 balloons that we put the 2-degree water in, and now we're putting the balloon itself into 2-degree water. Same thing for room temperature. We're putting the room temperature test tubes that we put into the balloons into room temperature water. And then that nice warm water as well. So what you're going to see on your sheet is this table here. And so we are looking at the change of circumference. And the circumference is the, if we were measuring around the balloon, we want to know, does that balloon expand, or is it going to shrink, or does it stay the same? What are your predictions? What's going to happen to those balloons?
They're all going to expand.
They're all going. Okay, so you're predicting that they're all going to expand, but at different rates and different amounts. You are right on track. Okay, so every 5 minutes for 30 minutes, that circumference has been measured. And that information has been put into this results chart for you. So what you're responsible for is determining how much did that circumference change. Okay, so you're going to go from 0 minutes here and look at your 30 minutes here to see what your total change is going to be. You're going to write this column of numbers into this column here on your graph. Answer the questions. And then what you're going to do is make a bar graph. So you have to interpret those results now. So if you go into your lab manual and you look here at Appendix A in the back, you're going to find how to draw a graph. And you can either hand draw a graph or you can do it online, or I'm sorry, in Excel. Either one is fine with me. Just make sure that you are putting a title on your graph and that you're naming the axes as well. So that is our lab. So you want to upload that to the lab, the post-lab questions. Okay. So any questions so far? We've only got a couple more minutes left, and I don't want to waste your time and go over either. Okay. So last thing I want to show you here on, well, I have 2 things I want to show you. If you need to leave, just let me know, or you can just, so let me show you these 2 things. So here we have the Science of Brewing Beer, which is just a 4-minute video about home brewing of beer and what that fermentation looks like in action. And then here we'll have our discussion forum. And this is set up a little bit differently than our last session. So when you open it up, if you come here into your discussion forum, you're going to see 15 different questions. And so what I'm asking you to do is to, oh, it looks like we have a couple here. Great. Let's see. All right. So Yeltsin has already gotten into this. That's great. So I'm asking you to make an initial observation and putting together the concepts that we're talking about and then answering these questions. So if I posted, why is beer bubbly? And so then Yeltsin went through and explained why beer has bubbles in it. And so there's 15 different questions. You can choose whichever one you want, but you can only choose one that no one else has chosen yet. And I put the parameters of what my expectations are for your initial post here. And then by Sunday, I would like for you to interact with other people's initial posts. And I want you to look at another item that ferments and compare those two processes and see what that looks like, whether it's yogurt or pickles, overexertion of muscles, all kinds of good things. Okay, so that is our discussion forum, if that makes sense. Okay, and now I want to also bring your attention to this review folder. I have uploaded a bunch of files for you that have review questions associated with each of the chapters that are different from your clicker questions. Some of them may look similar to your reading quiz questions. So I just want you to see that those are there for you to start reviewing, so that if you have questions for next Tuesday, you can bring those with you. And then we have our semester project. And I have the document here that I want you to type directly into, but I also left this open so that you can refer to it as you have questions. And what I'm looking for here is that you tell us about this person who has contributed to science. Instead of saying a scientist, it's really important that we're looking at these folks as contributors, because you know, not all of them, we're approaching it from we're approaching the scientific fact from a scientific standpoint. So it's important to give some credit to all of these different perspectives that have enhanced the scientific point of view and scientific knowledge, body of knowledge that we have. So I think that the questions are pretty self-explanatory. And if you have any questions about them, please let me know. All I would like for you to do is answer in full questions, type them directly into the box. And then that's what you're going to turn into me. So the last day that you can turn this in is the day of your last test. But if you want to turn it in ahead of time and ask for feedback, I am more than happy to do that. So anytime you want to get this off your plate, please do. I also want to show you this extra credit opportunity. This is where you get a chance to be creative. You can present this information in a PowerPoint, in a brochure, in a song, in a video. You know, however you would like to present the information, you are very welcome to do that. I'll give you 10 more points. Okay, so please use your creativity. So where are all of these contributors to look at? They're here in this wiki. So, all right, we've already got two. Yes, I've already found this, so you're good to go. So go ahead and you can look through these folks and see if there's anyone that speaks to you, and you get started. And again, you can't choose somebody that has already been selected. Okay. All right. I feel like I really rushed through those last two things, but I am sure that you are ready to go. Does anyone have any questions for me?
No.
All right. If anything comes up, please let me know. You guys are doing great. and I will see you next week. Thank you all for your time.
Good night.
Have a great night.